My lab is working on the early steps of HIV replication, and HIV has a very particular property in these early steps, which involve until integration of the viral DNA into host chromatin. So a very particular property in the sense that lentiviruses can replicate in non-dividing cells. This mitosis-independent replication of lentiviruses, and among them HIV-1, implies that the reverse transcribed DNA have to be translocated through the only entry door of the interphasic nucleus, which are the nuclear pores. And on contrary, all other retroviruses, such as Moloney, avian leukemia viruses, and so on, spuma viruses, are strictly dependent upon mitosis to growth, because their reverse transcribed DNA cannot cross this barrier represented by the nuclear membrane, and these retroviruses, have to wait for the mitosis and the disaggregation of the nuclear membrane during mitosis to access mitotic chromosome and integrate, in fact, a few hours after metaphasis. So the way the viral DNA, the retroviral DNA access to the cellular chromatin implies a replication mitosis dependent or independent. Thus, the translocation of through the nuclear pore of the HIV-1 DNA is a key for mitosis independent replication. This is of major interest because the main target cell in vivo for replication of lentiviruses are non-dividing cells, in particular macrophages, which are in number much more infected than T lymphocytes in the case of HIV. Macrophages are even almost the unique cells in vivo that replicate ungulate lentiviruses, such as Visna and so on. Another important reason to study the early steps of HIV replication is that from this mechanism of early step, from entry to integrate, we can derive gene transfer vectors, which are becoming more and more popular in both basic science and therapeutic gene transfer. And these new gene transfer vectors derived from lentiviruses, mostly HIV-1, keep the ability of the parental virus to infect non-dividing cells. In this case, we are talking about transduction of non-dividing cells. Transduction of non-dividing cells allows to access for genetic modification to cells or tissue of main therapeutic interest, such as hematopoietic stem cells, brain, liver. As a consequence of high efficient gene transfer, a lot of models for human genetic disease have been complemented, and this translates into human clinical trials, which are currently ongoing in France, in Paris in particular, and they are already giving all the signs of success. For the same reason of transduction of non-dividing cells, these lentiviral vectors turn out to be very efficient vaccination tools. The same reason because the main target cell to address for vaccination, for T-cell-based vaccination at least, is the dendritic cell, the professional antigen-presenting cell, and this cell type is non-dividing. So due to the ability to transduce non-dividing cells, lentiviral vectors are very efficient vaccination tools. We are deeply involved in the lab in a therapeutic vaccination project against AIDS, and we already obtained a very, very promising protection results in macaque cohorts, and this work is translating into a human clinical trial which will be set up by Infectious, a biotech company in Pasteur. Most interestingly, this same vaccination strategy could also apply to therapeutic vaccination against cancer, infection by hepatitis C virus or hepatitis B virus, and even malaria. We obtained recently very promising results for a quantitative vaccine against malaria. So, of course, today I will focus my talk on the basic aspects of early steps of HIV replication, with a particular emphasis on intracellular transport mechanism, and in particular on the role of a structure called HIV-1 DNA flap, a three-stranded DNA structure created during specific steps of HIV-1 reverse transcription, which is implied in nuclear translocation through the pore. In general, I will also emphasize on our experiments which aim to directly visualize HIV-1 DNA within cells or HIV-1 replicative complexes. One have to understand that these experiments are in general very complicated to perform, because fortunately for us, HIV-1 is quite a poorly infectious virus, and in vitro we cannot obtain low MOI infection, meaning that there's very few complex within infected cells. And since the early set correspond to only incoming material in the cell, the direct observation of these complexes or visualization of viral DNA is often extremely difficult, experimentally speaking. So, first part, I will talk about the very intriguing mechanism implying a cis-active element, a DNA cis-active element onto the linear DNA of HIV into the mechanism of nuclear translocation. We showed previously that reverse encryption of lentiviruses differs from the one from oncoviruses by 2 additional steps at the center of the genome. Normal, let's say simple retroviruses, as we say, goes from RNA to double-stranded DNA, classical double helix of linear DNA. In the case of lentiviruses, a central initiation governed by the central polymerase track and a central termination sequence lead at the very end of the reverse encryption process of a small strand displacement event of precisely 99 nucleotides, which lead to the formation at the exact center of the unintegrated linear HIV-1 molecule of a 3-stranded DNA structure that we call the central DNA flap. Mutations in the cis-active sequences responsible for the central DNA flap formation, such as CPPT, lead to the formation of the linear DNA devoid of central DNA flap. The mutations severely impair virus infectivity. And to resume in a single slide 10 years of research, on contrast to wild-type virus, which linear DNA is almost entirely imported into the nucleus and integrate or circularize. In the case of DNA flap defective DNA molecules, linear DNA accumulates, is normally routed with normal kinetics and efficiency to the nuclear membrane, but the cycle stops here. There's an accumulation of flap defective linear DNA at proximity of the nuclear membrane, as we showed by self-fractionation, by direct visualization, by in situ fluorescence in situ hybridization, by fish experiments, where you can see here that these little fluorescent dots are single HIV-1 molecules. In the case of wild-type viruses, these, by confocal microscopy, these fluorescent dots, these genomes, are found predominantly within the nucleus, whereas in the case of DNA flap viruses, these linear molecules accumulate predominantly at the vicinity of the nuclear membrane. To more precisely understand the nature of the replicative defect of flap defective viruses, we perform in situ hybridization coupled this time with electron microscopy, in this case transmission electron microscopy. And we confirm that HIV-1 viruses, wild-type HIV-1 viruses, integrate throughout the nucleus within open chromatin region, exclusively within open chromatin region. Whereas in case of either vectors devoid of flap or viruses mutated in the flap, the linear molecules accumulate at the vicinity of the cytoplasmic phase of the nuclear membrane, but very few of them are translocated within the nucleus. As always, it's not a black and white mechanism in biology. There's a few molecules that enter the nucleus, but most of them stay at the cytoplasmic phase of the nuclear membrane without translocating through the nucleopore. So to understand at the mechanistic level this very intriguing nuclear import mechanism, of course, a 3-stranded DNA structure, the DNA flap, involved in a nuclear import of a viral DNA is a very unusual mechanism. There's no known cellular or viral counterparts. Therefore, we had to make some hypotheses, very putative hypotheses, and one of our favorite hypothesis was the fact that most probably, on contrary to what is written in most notebooks or reviews on retrovirus replication, reverse transcription cannot occur in diluted elements in the cytoplasm. There's often a step called post-fusion uncoating or decapsidation of the viral complex, which is the disaggregation of this capsid into the cytoplasm as a necessary event for initiation of the reverse transcription. On the contrary, we notice in vitro that the strong displacement events during reverse transcription requires a high stoichiometry of reverse transcriptase enzymes, and this high stoichiometry must be maintained by a structure around the DNA intermediates. And thus we thought that reverse transcription was occurring within an integral capsid core, which maintains the enzymes around the DNA intermediates. The problem is that this capsid is much too large to be translocated through the nuclear pore. So mitosis-independent replication of lentiviruses implies an extra step compared to other retroviruses, which is a maturation of this complex upon completion of viral DNA synthesis, when this cage is no longer necessary, into a complex which is called pre-integration complex, PIC, of a size compatible with translocation through the nuclear pore. And since the last event is a strong displacement event which led to the formation of the DNA flap, we thought that the formation of this DNA flap as the last event of reverse transcription could trigger the decapsidation, the encoding of the reverse transcription complex into the pre-integration complex. Well, in the first attempt to test this hypothesis, we followed the intracellular outcome of capsid HIV-1 proteins by the simple fluorescent confocal microscopy. And what we observed is that in the situation of wild-type viruses or vectors that do contain the DNA flap, we see very early a perinuclear ring staining around the nucleus with monoclonals against HIV-1 capsid. And this perinuclear signal is completely lost between 24 hours and 48 hours after infection. On contrary, in the case of flap-defective viruses or flap first-generation lentiviral vectors without flap, we saw that this perinuclear ring of capsid signal appears with the same kinetics, but it is totally stable. You can wait for 10 days, 15 days if you wish. You will still see this perinuclear ring around the nucleus of capsid signals. To visualize directly the nature of complexes associated with this capsid signal around the nucleus, we first perform transmission electron microscopy on infected cells with wild-type or DNA flap-defective viruses. We never see any structure with wild-type viruses, but with flap-defective viruses, we see very occasionally, I have to say, typical conical-like structures that resemble a lot to HIV-1 capsid in length and in shape. It is very close to what is described in the extracellular particle. You see here this core of conical shape, and here is very reminiscent of this structure. These capsid-like structures are always found at close proximity of the nuclear pores. You see here the invagination of the nuclear membrane. There's two pores, in fact, nuclear pores here, and in front of the pore, these structures are sometimes found. It's very rare, in fact. The viral nature of this structure has been confirmed by pre-embedding capsid labeling with immunogold antibodies, and a very heavy labeling is seen of this capsid, confirming the viral nature of these structures. So, of course, by transmission electron microscopy, and because multiplicity of infection with HIV is very low, it is extremely rare to have the chance to cut in a longitudinal manner a capsid in infected cells. And so this experiment has to be taken not in a qualitative manner, I would say. To more precisely quantify this phenomenon of capsid accumulation in case of DNA flap defective viruses. We perform surface imaging of nuclei, of infected cell nuclei by doing scanning electron microscopy of infected cells. You see here the nucleus, the surface of the nucleus of a HeLa cell is a lot high density of nuclear pores, a little holes here, and actin filaments surrounding this nucleus. In infected cells, again at late time points, only in the case of flap defective viruses, we do see exactly on top, well, onto the nuclear pores, we do see capsid-like structures which are each time onto the nuclear pore, but slightly excentered from the lumen of the pore. And capsid gold labeling showed a very heavy staining, confirming the nature of the viral nature of this structure. We do see also at proximity of the nuclear pores on these actin filaments that surround the nucleus, capsid structure, again heavily labeled with immunogold, anti-capsid immunogold antibodies. And this time we could quantify precisely the labeling per surface of the nucleus, and this situation again is seen only in the case of flap defective molecules. Of course, the labeling with immunogold perturbs the ultrastructural, the shape of the capsid, because there's a lot of antibodies with 10 nanometer gold beads which bind to the capsid, and to have a more precise idea of the exact structure of this capsid, we perform cryo scanning electron microscopy this time, which we know preserve intact the cellular ultrastructures. By cryo scanning, we do see exactly the same picture as a nuclear membrane with actin filaments and nuclear pores, and sometimes on again slightly excentered from the lumen of the pore, we do see bullet-like structure, which are exactly what is expected in case of HIV-1 capsids. Are artificially colorized in blue here, and you see that they are just interacting with nuclear pores underlying this capsid in green here. So the conclusion of this first part is that the formation of the central DNA flap as the last event of HIV-1 reverse transcription facilitates the maturation of the reverse transcription complex, which is in fact directly derived from the core of the particle into a pre-integration complex of a size compatible with nuclear translocation. First, other immediate consequence is that there's no post-fusion event as we thought before. This uncoating step occurs at the nuclear pore upon completion of viral DNA synthesis. And we cannot exclude, in fact, we have indirect arguments that upon accessible to karyophilic proteins of viral or cellular origin, the DNA flap is also involved in the translocation itself, allowing the crawling of the filaments through the nuclear pore and the subsequent integration. Well, HIV-1 nuclear import is not only nuclear translocation through the nuclear pore. This process must involve a lot of steps, and among them, the routing of the HIV-1 capsid from the plasma membrane to the nuclear plasma, to the nuclear membrane. Sorry. And to observe these steps, we set up a technique allowing for real-time imaging of HIV infection in living cells by tagging the integrase, a C-terminal tag with a small peptide, which is a tetracysteine tag described by Tien. And this tag allows the binding with a high affinity of a bisarsenical derivative of fluorescein, which is called a flash. And this small tag do not, as we will see, do not perturb the HIV-1 infectivity. The problem of tagging viruses, in particular polyproteins of retroviruses, is that most of the modification have a pleiotropic effect and lead to non-poorly or even non-infectious viruses. Using this small tag, first, the staining was perfectly specific of the integrase, but also, and most importantly, infectivity of the tagged virus and stained virus with flash was fully retained, was intact. That was the same infectivity as wild-type virus. So, by collaboration with Dr. Olivo Marin Laboratory, we perform real-time studies, and you will see that the routing process involves, in fact, several very different types of movements. And by the use of a 4D dot tracking software, 4D meaning 3D and plus time, an automatic software, we could follow hundreds of trajectories and analyze hundreds of trajectories within the same acquisition. So you can see on this video, there's a 2D reconstitution here and a 3D reconstitution here. That is it working? Yes. That routing from the plasma membrane to the nuclear membrane involve first a very quick and saltatory movement with peak velocities of up to 1 micrometer per second, which is strongly reminiscent of a microtubule-dependent movement, but close to the proximity of the nuclear membrane at 200, 300 nanometer. The viral complexes undergo a transition from this very quick transport to a much slower movement and more curve, which lead to the docking of the complex onto the nuclear membrane. And once docked, in fact, we still see movement in the range of 25 nanometer per second, but in a very confined volume, a sort of vibration which persists a very long time for hours before translocation. The overall process is very quick, a few minutes, but the docking stage is extremely long. So the movement characteristics are strongly reminiscent of both microtubule followed by actin-directed movement. The implication of microtubule and actin was confirmed by both colocalization experiments with microtubule and actin filaments surrounding the nucleus, and specific inhibition experiments using the CC1 transdominant negative domain CC1 of dynactin and latranculin B treatment for actin movement. Using a tag in the integrase, another advantage is that we can follow the infection from entry until integration, because integrase is, of course, the only protein which is for sure involved until integration of the viral DNA. There's around 100 molecules tightly bound to the linear DNA, per linear DNA. And integration should translate by the sudden disappearance of fluorescence, since the formation of oligonucleosome will chase the integrase, the viral integrase molecules. And that's exactly what we observe times 2 times. You have here 3 intranuclear complexes, which are represented in 2D here and in 3D here. Only very slow and diffusive movements are found within the nucleus, but occasionally you see dots in which fluorescence suddenly disappear. It's not quenching because you have the 2 brothers which do not quench at all. So this is most probably, we don't have the direct evidence, but this signs most probably an in vivo integration event. Well, altogether, this data represent a profound revisitation of the early steps of HIV replication. Once again, there's no post-entry fusion event, uncoating event for reverse transcription. On contrary, fusion of the particle to the target cell leads to the injection of this core of the particle, which is in fact the reverse transcription complex. The intact capsid shells are rotated via first in a rapid manner via microtubule, and then at the proximity of the nuclear membrane via the transition onto actin filaments to a nuclear pore. You see that you have a perfect concordance, a perfect. We see these complexes at the electronic microscopy level, but also in real time. And these once docked at the nucleus, this step is very long, and reverse transcription requires 10 to 12 hours, whereas rotting requires, let's say, 10 or 20 or 12 minutes. So we think that most of the reverse transcription process occurs within this intact capsid shell, which are already docked at the nucleus, not at the cytoplasm, as classical review say. And once HIV DNA is reverse transcribed, and upon completion of viral DNA synthesis and DNA flap formation, there is this major morphological change, which is the maturation of the reverse transcription complex into a pre-integration complex that will be able to be translocated through the nuclear pore and subsequently will integrate or circularize. Well, I have to thank my collaborators of the lab, and most particularly Nathalie Harel, which did a lot of the experiment that I presented today, and also my collaborator, Spencer Short, with whom we worked a lot with Spencer Short for the setup of this experiment, also with Jean-Christophe Oliveau-Marin for the quantitative aspect of 4D tracking software. And Terry Ellen in Manchester, we went for learning this scanning electron microscopy techniques. It was at that time the only lab in the world to be able to do this kind of technique. Thank you very much.
Are there any questions? Maybe I can start. I mean, with regard to this, the fact that reverse transcription occurs in this confined environment of the capsid, do you see any rationale for this? Are the other retroviruses acting similarly? I'm just trying to figure out why it should have to remain within these limits, maybe for efficiency purpose.
Well, in fact, other retroviruses behave the same. We know now quite recently that in fact what is imported after the mitosis in Moloney viruses, for instance, is an intact capsid which will participate until the integration process of the oncoretrovirus. But for lentiviruses, this capsid cannot go through the nuclear pores; they are too big. So there's another challenge as additional steps which involves this maturation of the capsid. So now why is there the necessity to perform the reverse transcription process within a confined volume? It's simply because the enzyme, the HIV1 reverse transcriptase enzyme is very distributive, meaning that the enzyme will drop very often from the DNA intermediates, and if you don't have a cage, a protein cage that will maintain the enzyme and allowing for re-entry of the reverse transcriptase to go on the process of reverse transcription, it's hopeless to have a complete reverse transcription.
Okay, thank you.